The regulation of gene expression in multicellular eukaryotes especially is so complicated that it's easy to be distracted from the main point. Regulation is about the specificity of interaction between regulators and the genes or gene products that they regulate. In multicellular organisms, different cell types are different even though they all have the same genes. What distinguishes the cell types is that they use their genes differently. For example, muscle cells and cells in the retina make some of the same proteins and RNAs and some distinct ones. Gene expression in eukaryotes may be regulated at many levels. For example, in a skin cell, a particular protein encoding gene may or may not be transcribed, and if it is transcribed, the primary transcript may or may not be spliced and otherwise processed, or it may be spliced in a different way in skin cells than in other cell types. If an mRNA is produced, it may or may not be exported to the cytoplasm for translation, and if it is exported, it may or may not be stable in the cytoplasm, and if stable, it may or may not be translated. The protein product can also be regulated. Amino acid modifications can destabilize the protein, localize it to a particular subcellular region, or affect the protein's activity. Let's preview just a few of the examples that you will soon see. In fruit flies, the double-sex activator protein is made in females and binds an enhancer of the yolk protein 1 gene, YP1, activating its transcription. In male fruit flies, the male form of double-sex is made and it's a repressor. When the double-sex repressor binds the enhancer, YP1 is not transcribed. These two double-sex transcription factors are each specific for a limited number of genes, including YP1. The amino acid sequences in the DNA binding domains of these proteins are able to make attractive contacts with specific base pair sequences in an enhancer of YP1 and in the enhancers of certain other genes. Transcription factors, activators and repressors, regulate transcription initiation of specific genes. They interact only with particular genes because enhancer binding requires specific protein DNA interactions. Female and male fruit flies express different forms of the double-sex protein through regulation of RNA splicing. The primary transcript of the double-sex gene is made in both males and females. However, only females make transformer, TRA, a protein that regulates splicing of double-sex RNA. In females, TRA binds the double-sex primary transcript at the splice acceptor site of exon 4. Only when TRA is bound there is the fourth exon used. In males, because TRA is absent, the fourth exon is skipped. Regulation of RNA splicing is how males and females make different double-sex mRNAs and therefore different forms of double-sex protein. Amino acids in the RNA binding domain of TRA protein are attracted to a specific base sequence in double-sex RNA. Transformer regulates the splicing of a limited number of primary transcripts, including double-sex, through specific protein-RNA interactions. After splicing, mRNAs can be regulated by microRNAs, small RNAs that join a protein complex called RISC. MicroRNAs bring the RISC complex proteins to specific target mRNAs through complementary base pairing between the microRNA and the target. The result is either degradation of the mRNA target or inhibition of its translation. Here, the specificity of the regulation comes from RNA-RNA interactions. The base sequences of microRNAs are complementary to their targets, allowing for base pairing interactions. One final example. The ribosome will bind to mRNAs only when they are in a complex that circularizes the mRNA, which includes a protein called 4E. Formation of this protein complex is inhibited by a protein called 4E binding protein, which competes for 4E binding with other proteins in the complex. When 4E binding protein is phosphorylated, it cannot bind 4E. Specific kinase proteins phosphorylate 4E binding protein, and specific phosphatase proteins remove the phosphates. The higher the fraction of 4E binding protein that is phosphorylated, the more mRNAs will be translated, and the lower the fraction, the less translation will take place. Therefore, the kinase and phosphatase proteins regulate translation through their specific interactions with 4E binding protein. Amino acid sequences in specific domains of the kinase and phosphatase proteins are attracted to certain amino acid sequences of 4E binding protein. You can see that specific protein-protein interactions can also regulate gene expression. Remember, regulation is all about specificity of so-called regulatory molecules for particular genes or gene products.